Good morning, good afternoon, and good evening, everyone, depending upon where you're located. My name is Sarah Jane Demy, and I am the founder and CEO of Demy-Colton, and it is my pleasure to welcome you to the fourth edition of the Demy-Colton Virtual Salon Series. As many of you may know, Demy-Colton's mission is all about convening biopharma and digital medicine executives to catalyze change. We know we can help drive innovation when we bring individuals together who are united by a common cause and a shared passion. And in our industry, that cause and that passion is bringing cures and therapeutics to people in need. We were in the process of launching a new business development and investor conference here in New York City named BioFuture when COVID-19 interfered with those plans, as it did with the plans of people all around the world. So in lieu of an in-person event, we launched the Virtual Salon Series to continue to bring great content to you and continue our mission of bringing people together. Sorry about that. Before we move to the formal portion of our salon, I want to recognize and thank all of our sponsors who have stood with us during this time and indeed have broadened their support as our programming has evolved. We will continue to bring you essential and thought-provoking discussions virtually on a regular basis. Please check our website for the schedule of salons, topics, and participants, and as they will change and it will be updated on a continuous basis. Today's installment of the Demy-Colton Virtual Salon Series is in vivo gene therapy, personalizing ultra-rare and scaling up for chronic disease. Before I introduce the moderator, please take a moment to review the forward-looking statements. As always, the panelists' remarks are their own and should be treated as such. The discussion will be led by Mike Rice of Cello Health Bioconsulting, and he will be joined by Faraz Ali of Tenea Therapeutics, Sapan Shah of Stride Bio, and Jeff McDonough of Generation Bio. We do hope that all of our panelists will join us when we finally do our in-person meeting, which is currently scheduled for April of 2021. And I think the jury is still out about whether or not that will actually happen. Let me introduce Mike now. Mike is with, as I noted before, Cello Health Bioconsulting, where he leads the gene and cell therapeutics and rare diseases practices and co-heads the oncology practice, focusing on hematological malignancies and genetically defined cancer. Prior to Cello Health Bioconsulting, Mike gained over a decade of experience creating new biotech ventures from academic inventions pertaining to nucleic acids and cellular platforms applied across monogenetic diseases and oncology. Industry positions have included translational research, strategic planning, technology transfer, finance, and business development. Now, today's virtual salon will be structured a little bit differently than what we've done in the past. At the conclusion of the formal portion of the discussion at about 11:50, we will move to a live 15-minute Q&A session with the panelists. We encourage you to stay if you can and submit your questions in the lower left-hand corner as we go on. Now I'm pleased to turn the discussion over to Mike and to our panel. Mike, take it away.
Yes, thank you, Sarah. And I really want to thank the Demy-Colton team for inviting me to moderate this great panel. This topic is very, near and dear to me. As you mentioned, I've spent over 20 years, as an inventor, a entrepreneur, and advising gene and cell therapy developers how to extract value from their platforms and position indications that they can develop themselves and things that they can think about, to license or partner out to developers that, could go beyond what a small biotech could do. The advanced therapeutics practice at Cello Health Bioconsulting basically does this on a daily basis. We work with both developers and the in-licensors and acquirers to help inform therapeutic strategy but also with diligence for in-licensing assets and thinking about how does it fit into, say, a large pharma portfolio. I have some material. I want to get into a little bit of the background for this topic and set the stage for the panelist discussion. But I have a companion sort of handout that if you email me after this this webinar, I can distribute it, or we can send it through Demy-Colton. But really, from working with biotech developers as well as large acquirers of gene therapy technologies, it is clear that it's found its place in the continuum of the evolution of therapeutic modalities, from small molecules to biologics to nucleic acid therapeutics and ultimately regenerative medicines. And regulatory agencies are now evaluating gene therapy programs merely based on the therapeutic potential. So that said, there's a lot of progress going on in both the advancements of the evolution of platforms, but also thinking about how do we review these for registration, how do we think about reimbursing one-time curative treatments, and addressing, in some cases, very complex logistical manufacturing. So to give you a little bit of a view of where we are right now, there's, this has been a very attractive area for investment, particularly early stage investments. There's been over $4 billion worth of venture financing and IPOs since January 2019. That's only a year and a half. And this year already, the first 7 months, we're already approaching $2 billion. So it's quite an interesting time period, particularly with what's going on with COVID-19 and the lockdowns and everything. So there's been a lot of capital moving into the space. And the reason why it's been very attractive because, there's been a high success rate of translating new inventions into therapeutic potential in the clinic. And there's been $34 billion in exits through acquisitions since the seminal Avexis acquisition by Novartis for $8.7 billion in 2018. So we'll see what the rest of this year has in store for it as far as investing and what sort of acquisitions. There haven't been any major acquisitions really this year, but there's still 5 months left to go, and we'll see what happens as these inventions move in the clinic. So since today we're talking about the healthcare delivery, the future of healthcare delivery, we can talk about many topics because it's a very broad technology space. But we really want to focus on in vivo gene therapies that are more off the shelf, they're more drug-like, and think beyond the potential for where they are right now in ultra-rare diseases and how they might be applied to more prevalent chronic disorders. So that first generation gene therapies are now validated in many different monogenic diseases, at least, and even commercialized, such as Zolgensma for spinal muscular atrophy, Luxturna for RPE65 deficient inherited retinal dystrophies, and as well as many other ex-vivo approaches or engineered cells that are been driving the adoptive cellular therapy space. But we really want to focus on the in vivo applications. There are 410 in vivo assets in development from preclinical to phase 3. 114 of them are in the clinic. So it's a very active area for clinical translation, clinical development. 25% of them are pursuing non-ultra rare diseases. So that's a pretty big component of them are already, pursuing more complex disorders. By 2026, Evaluate Pharma forecasts $14 billion of worldwide revenue from in vivo gene therapy. So from where we are now to where we may be in 5 years, could be a really incredible transformation of delivery of healthcare approaches. So in some cases, these are providing new therapies for neglected, diseases where there are no other treatment options. Other cases, they're coming into mature markets that, may obviate the need for protein replacement therapies, such as clotting factors for hemophilia. We're closely following BioMarin and their Veloctogene program and what might be happening there. There'll be great case for transformation of healthcare if it plays out the way we see it. And also, one by one, across the entire, enzyme replacement therapy field, there's very active development of programs pursuing them. So these in vivo gene therapies are now beginning to expand beyond the ultra rare into more prevalent diseases that have potential near-term approvals, even in things such as ophthalmology. Age-related macular degeneration, neurodegeneration associated with Parkinson's disease, and certain cardiovascular diseases such as cardiomyopathies and dysrhythmias. So we're going to see a lot of advancement, ultra rare, but also the potential is going to be applied to these, more broader disorders. So today's panel, we're going to discuss the state of the art of first generation therapies, what we've learned from them, and then what improvements are needed to, make these therapies more broadly accessible to more common chronic diseases. And so, we're going to discuss topics such as, investment risk, going beyond the ultra rare, transformation from the one and done approach to repeat administration. What sort of progress is really there to change it for these more chronic, complex disorders? What needs to change in the safety bar to make it acceptable for less severe diseases, different therapeutic strategies, commercial strategies. There's a lot of topics for us to get in today. So let's just jump right into it. So I would like the panelists to actually, go a little bit beyond Sarah's introduction and talk a little bit about, their background, what they're doing now, and some of the prospects they see for their platforms that they're developing. The panel was selected based on diverse approaches of in vivo approaches, from AAV gene therapy to non-viral gene therapies and other sort of engineered capsids that go beyond what is currently available with first generation vectors. So let's get into it. So Faraz, why don't you start? Just give a introduction of yourself and what you're doing at Tenaya.
Hi, thanks, and thanks for the opportunity to be part of this this great panel. I'm the CEO of Tenaya Therapeutics. I've been there for the last 2 years. Previous to that, I was at REGENXBIO and Bluebird Bio, so I've spent the last 10 years in gene therapy. And prior to that, was at Genzyme Corporation for almost 10 years, where I had the great privilege of launching products for several rare diseases, but an even greater pleasure of actually working for Jeff McDonough, who's a fellow panelist here today. Tenaya Therapeutics is a company advancing next generation therapies with a 100% focus on heart disease. We have 3 product platforms, including gene therapy, but also regenerative medicine approaches and precision medicine. We're technically modality agnostic, so we are a lot of what we do is gene therapy, but that's not all we do. We actually have some very promising small molecule work going on in-house. We target both orphan and prevalent conditions, and in fact, part of our strategy is to start and assess the efficacy and the safety of a target in a rare population before moving on to a larger population. A private company, preclinical stage, in IND-enabling studies for our first asset that's making its way towards the clinic. And we have just 65 very passionate people, all located in South San Francisco.
Great, thank you. Sapan, do you want to go next?
Yeah, sure. Thanks, Mike, for the introduction. I feel like I've been in a staring contest with my webcam and losing, together with Jeff and Faraz, trying to piece it together. But appreciate you bringing everybody together in this format. It's nice to kind of engage in this way. My name is Sapan Shah, CEO of Stride Bio. We're another next generation gene therapy company based in Research Triangle Park, have about 53 employees. Our piece of the puzzle is we're trying to improve the vector, so the delivery system that is used to make these gene therapies possible. And our unique piece of it is we focus on the 3-dimensional structure of the viral capsid and use our knowledge of that to identify the epitopes and regions that we want to modify and create diversity around, and thereby build in novel properties into that delivery vehicle. So things like evasion and neutralizing antibodies, specific tissue tropism, enhanced manufacturability and improved potency. We're taking those novel capsids and combining them with payloads of interest. We have 3 really amazing partnerships, one with CRISPR Therapeutics, one other with Takeda Pharmaceuticals, and a third with Sarepta Therapeutics, where we're working together to advance programs in novel areas of interest. My background, I've spent the last 20 years as a CEO of multiple different companies, large and small. Been with Stride Bio for about 2 years, but have spent prior to that a long time on the sort of conventional therapeutic side. So it's interesting for me to see some of the differences between what we do in the small molecule space versus what is the promise of gene therapy, but then also the challenges, which I think we'll talk about. My background is I have a PhD in molecular biophysics and biochemistry. And so started early on thinking about structure function. And so have come full circle a little bit with how we're doing that with AAV at a company founded by Arvind Asokan and Mavis McKenna, who pioneered a lot of the work and our knowledge of the structure function of AAV. And after getting my PhD, spent a number of years at McKinsey & Company advising large and small companies in the pharma and life science space. And so really thrilled to be a part of an innovative company like Stride. Thank you.
Great. Thanks, Sapan.
Yeah.
So Jeff, you want to go next?
Sure. Thanks, Mike. And thanks for those great intros for us and Sapan. So Jeff McDonough, CEO of Generation Bio, was CEO of leadership from BioFuture for 6 years before this and spent a decade at Genzyme with us and many other incredible people during an amazing time for rare disease biotech. Grew up and trained in the Harvard system as an MD prior to that, so a winding path. Generation Bio was specifically founded to create a new class of non-viral gene therapy, durable, redosable, and scalable to address millions of patients living with both rare and prevalent diseases. As a non-viral platform company, it's really looking to integrate 3 novel technologies to try to build on the very obvious demonstrated success of first-generation AAV gene therapy. Specifically, a novel DNA construct called closed-ended DNA, which translocates the nucleus and yields the same durability of expression as AAV does in the nucleus. Second, a cell-targeted LNP delivery system, which yields the redosability and very specific biodistribution properties of our delivery. And finally, a capsid-free manufacturing platform that allows for our construct to be manufactured on traditional biologics infrastructure. That really opens up, as Faraz was saying, this pathway from rare to prevalent indications. We have 8 programs, 5 in the liver, 3 in the retina, and 3 expansion areas that we're looking at, including skeletal muscle, central nervous system disorders, and oncology. So a broad kind of platform vision, but really centered around integrating these 3 technologies to try to build a non-viral class of gene therapy. So it's like to be here.
Great. So thanks, Jeff. And I think you gave a good segue to the first question. And I just want to remind the audience that we are fielding questions. So we're going to have a little bit of a guided discussion, but towards the end of the hour, we will answer any questions that we can get to. So please send them in to Sarah. So anyway, so the first question is for Sapan. And since Jeff mentioned the progress of first-generation gene therapies, and that they're real, they're here. We have approved products, and many of the things that are just entering the clinic right now, as we know from experience from say Zolgensma, is that you can go very quickly from a phase 1/2 program with a small cohort of patients, all of a sudden thinking about how are you going to commercialize this thing. So what have we learned from first generation, or what are we learning from first generation gene therapies, and how do we think about how to go beyond that?
Yeah. Thanks, Mike. No, I think, just thinking about your opening remarks as well, what an incredible time for the field and for patients. And it's just a staggering number of programs. Hearing you say it, it just drives it home how much activity there is and how quickly the pace of innovation is in this area. And quite amazing also to think that there's just 2 approved in vivo gene therapies to date. But what those two programs and the teams at Avexis and Spark have done is really prove the concept for us, right? I think what captivates investors and why Jeff, Faraz, and I are probably in this space as well is just the opportunity that those initial approvals represent. And the concept is you can take, in the case of in vivo gene replacement, take a healthy copy of a gene and deliver that to a patient with a mutated or diseased version of that gene. And with a durable expression of that construct, have, as you said, a potential for a once and done therapy. And, when I explain that to people, particularly lay people who aren't in the field, I mean, they just sort of like, is that really possible? I mean, can you really do that? And it's like, well, yeah, we have two companies that have gotten that to the finish line, and that paves the way for us to think about next generation therapies. So, I mean, one of the challenges here relative to conventional therapeutics is that while you are delivering a replacement copy of a healthy gene, it's a very complex modality. And if I think about the treatments that we're advancing, there's really every gene therapeutic has three components. Really, it's like three products in one. You have the delivery vehicle, you have the payload of interest, and then you have the manufacturing process, which I think in the gene therapy space is part of the product. The process drives the product, and the product drives the process. And so the innovation that you're going to see is, I think, going to be around each of those three components. And we've started out at Stride Bio trying to improve the vector. And I know Faraz mentioned some innovation around the payload, and Jeff as well in terms of non-viral approaches. So I think to expand beyond these monogenic settings, you're going to need to innovate around all three of those and bring these therapies to a broader cohort of patients. But what's been done to date, I think, has proven that it's doable. And, it's up to sort of these next generation companies to kind of take that beyond what those initial pioneers and innovators have done. So I'll pause there, see if others want to chime in.
Great. Anybody want to add or comment to that? No? Okay. So for us, we've set the stage on what is, currently, going on and the rapid progress. I did mention that 25% of the pipeline that's in the clinic already is, that might be including preclinical, but a significant number of programs are pursuing broader disorders. Probably firstly led by ophthalmology indications such as age-related macular degeneration, but also forms of, CNS, small, like a rare subset of Parkinson's disease. They might be the first approvals. So what is the potential to go beyond the ultra rare, and think about broader applications? I know you have a focus in cardiac disease. So, you're going to be leading the charge, I think, in some of the really broad potential of addressing some of the highest unmet needs, cardiovascular disease.
Thanks for the question. And well, first I want to give credit where credit's due. I mean, there were other pioneers who kind of created the vision here. In wet AMD, as you mentioned, I had the privilege of working at REGENXBIO, and that, in addition to the orphans, they added that to the portfolio. And that's not in a situation where you're adding a healthy copy of a gene, but actually vectorizing a fragment of an approved biologic against a validated target. I thought it was very clever what they did, and that shows the potential to go beyond rare genetic disorders. And so now there's others in that space as well. In the heart disease, actually, we're also not the first. Celadon as a company that had a go at this a few years ago, and they had a good run of it until the phase 2, and we learned some things from that process. We learned that the vector selection has to be good. They were using first generation vectors, and we now know we can do better. And there were things that we learned from that experience about manufacturing and the importance of getting manufacturing right. So in order to go from orphan to prevalent, which is something we think a lot about because, as I said, our explicit strategy is to start an orphan and then expand from there. I would say that there's, in the near term, two things that we just got to make sure we get right. And one is safety and the other is manufacturing. And on the safety front, similar to Sapan and Stride Bio, we think a lot about the vectors. And so we have internalized in-house the ability to create novel vectors with specific tropism for the heart. And even within the heart, we work on trying to find ways to more selectively target specific cells and more selectively express the gene of interest in, say, a cardiomyocyte versus a cardiac fibroblast. So getting to that level of specificity, I think that's it. You know, the orphan market with very severe patients, that's a more permissible environment. But when you try to go broad, safety is going to be paramount in the mind of the regulators and patients and physicians, as it should be. So we do a lot of work on vectorology and promoter to make sure that we can detarget the liver, for example, or detarget the brain with our vectors. And so, like Stride Bio, we're not. We think there's a limit to how far you can go with the existing vectors, the parental vectors, and so we've made an investment there. Manufacturing, I guess, has become the Achilles heel of the space. Often people are focused on that through the lens of, you know, the amount of dose that you have to produce, and also through the bottlenecks at CDMOs. But what we really are trying to think about from the get-go, really, even when we were 3 or 4 years away from the clinic, now we're about 1.5 years, we were already beginning to think about how do we get to a point where we can scale up these products to not just serve a few thousand patients, but potentially to serve millions of patients. And we knew that we would need breakthroughs in manufacturing to get there, and we have been working on those and internalize that. And we've, in fact, recently filed IP in manufacturing for AAV. But we need to scale up partly to be able to produce drug reliably to serve large patients, but partly to bring down the cost of goods and bring them to a commercially reasonable place where they can compete with traditional biologics, and where you know, we won't be having the same conversations we have about price that is often plagues the orphan market. So I think safety and manufacturing in the near term. Long term, I also think financing strategies have to be thought through. When it's a one and done, theoretically, the payers have shown a certain amount of uncertainty or discomfort with the uncertainty of, well, will it really last all the years that you think it's going to last? So I think early on, there's already been some signals of a willingness to engage in risk-sharing agreements when there's so much uncertainty about the long-term safety and efficacy of a one-and-done therapy. And I think that's welcome. And that's welcome in traditional therapeutics, but definitely welcome in potentially curative gene therapies. So safety, manufacturing, and financing, I think innovations in all of these domains will be necessary to allow for to grease the tracks, to allow gene therapy to go from rare to prevalent.
Great, and great for us for bringing up, you know, the reality. We jumped in a lot of the hype and the hope around gene therapy, but, you know, safety and adverse events, are still an issue in certain programs, and everybody, I'm sure, has been following the news around Densi's XL-MTL program and Solid Bio's Duchenne muscular dystrophy program, particularly, high titer, some of the issues with high titer viral particles and other things. So, the reality is we need to address those. It brings to me some parallels to the RNAi or the antisense field, a nucleic acid therapeutic field, where we have now, I think, 5 or 6 programs for Duchenne, for SMA, for rare hepatic disorders that have been approved, both antisense and RNAi, and in many of those cases, there were some programs that were discontinued because of adverse events. And when inclisiran came around for a broad cardiovascular program targeting PCSK9s, there was a big haze, initially around, can we move this to a broad patient population, given, the hepatic encephalopathy, the innate immune activation, other things that we've seen in the field. But, I think that is soon going to play out, and there'll be another example of how a new modality has transformed the field, but that has yet to play out. But so I guess following up on that, I guess, Jeff, I'd like to hear, from you, what, improvements, are going beyond first generation gene therapy programs, and what is needed, beyond where we currently are to address these broader therapeutic areas.
Yeah, thanks, Mike. I mean, maybe I'll take a crack at that in 3 dimensions, because I think each of Faraz and Salvan and I see that in very similar ways. The construct, the method of delivery, and manufacturing are all integral components of either defining or expanding the reach or potential of gene therapy. Maybe just a quick comment on each of those in that case. So As it relates to the construct, one of the major limitations there is the size of the gene that can be included inside the viral capsid. It's currently limited at 4.7 kb, and that's a physical limitation of the capsid structure itself. What that means is for a given gene that can fit within that size, you're forced to optimize and minimize the size of the regulatory elements that are around that gene. So the ability to take advantage of intranuclear dynamics around expression is limited by that capacity limitation. It also limits the number of genes or the number of diseases that can be addressed within that finite cargo capacity. You know, when we think about expanding that dimension, it's not only the ability to address new diseases that have genes that are too large to fit in traditional AAV capsid, but also the opportunity to think about polycistronic therapies where you can express more than one reading frame in a single construct. That opens up a very interesting space of creating antibodies, for example, against multiple targets on a single construct, or polyfunctional enzymes or other proteins. So I think that dimension of the construct is one place where lots is being done within the AAV field. And as you start to open up the capacity to making constructs in the 12 to 15 kb size, you get lots of opportunities to play in existing and also in new diseases. I think delivery is a very fertile area. You know, I think where AAV works today from a delivery perspective, it is highly effective. And tropism is an issue, as Faraz is saying. I mean, in some of these high titer examples you gave as well, Mike, we're delivering 100 to 1,000 viral copies or capsids for every cell in the human body, only to reach, you know, a few, relatively in proportion, a few cells in the target tissue of interest. So this balance of on-target and off-target is something that has plagued the AAV field for a long time. And so there's lots of room for that to improve. I think one of the broader issues, though, that's maybe bigger than that is pre-existing immunity against capsid, which patients can carry from their natural exposure to AAV. That excludes many from getting the benefit where AAV has proven to be effective. So I think conquering that is a big place for potential improvement. And then this one and done, I mean, in post-mitotic tissues, it's probably true that one and done can work. However, when tissues are mitotic at the time of therapy, one and done probably isn't feasible. And when you're limited to a single dose, it means that it's impossible to individualize therapy. So some patients are too low with a single dose, some are just right, and some are too high. But it's hard to predict that up front. And once you've got a certain level of expression or not post-dose, there's nothing you can do about it. So I think what we're seeking in a variety of technology approaches is to get more individualization to make gene therapy more drug-like. I think that's an aspiration that all gene therapy and genomic targeted companies have in common. And once you get redosing opened up on a sort of easy, low threshold basis, it also allows you to treat kids. And I think one thing that Faraz and I learned over and over and over and over again at Genzyme is that the healthiest day of a child's life with an inborn error of metabolism is the day they're born. And every hour and every minute that passes after that, they lose something. So I think for gene therapy to really make a difference in genetic disease, you'd ideally like to be able to get in and intervene in the infancy or neonatal period. Hard to do that without the ability to redose when the target tissue is still dividing.
Can you explain that maybe a little bit further for the audience? So if you have a AAV9, vector for a particular, genetic disease that's available, shortly after birth, I mean, why is retreatment, essential there that you're saying?
I think it may not be essential when the target tissue is not growing, Mike. So when the volume of target tissue, as it is in the case of the spinal motor neuron axis, I think a single treatment will work and is likely to be sustained for as long as the cell lives. I think when the target tissue is the liver, which is highly mitotic, even in adulthood, but doubly, triply, quadruply so in early childhood and development, because gene therapy in the nucleus doesn't divide and go to daughter cells. As those cells divide, the relative dose of your gene therapy gets diluted. So I think you want the opportunity to redose for effective pediatric treatment.
You're right. And reminding everybody that the approaches I think we're talking today are all episomal, right? So they're not integrating in the chromosome. Potentially down the road, if gene editing, in vivo gene editing becomes, reality, that could potentially be a solution to that.
Yeah, and as for us, as all of these things are going to be held in a balance of safety and efficacy against the relative severity and urgency of the disease, right? And just to take the last aspect here, I think overcoming this limitation to rare disease is important from the point of view of manufacturing and scale, because access is limited today primarily because scale is just very difficult to achieve with viral manufacturing. And as Faraz is saying, breakthroughs are needed there. And alternative approaches like ours, among others, that are not producing viral capsid have the potential to really help boost or contribute to that journey from rare to prevalent diseases. And just to take a payer point, which Faraz rightly focuses on, payers would appreciate a drug like gene therapy also. They don't mind paying for therapy multiple times if that multiple dosing allows for both individualization of treatment and prolongation of the benefit. So I think redosing and bringing COGS into the right range and allowing us to approach really prevalent diseases are all key places where we can make progress.
Great. Yeah. All right. Well, I think we touched on a bunch of topics. I'm sure the panel wants to jump on in there, from immunogenicity to prior AAV serotypes to manufacturing. Who wants to jump in on that?
I'm happy to. I'm always happy to follow Jeff. So you made a lot of great points there, that interplay between the construct, delivery, the actual organ. So there's not going to be one sort of silver bullet here that's going to work for every indication in every therapeutic area. And so the dynamics are unique. In our case of the heart is post-mitotic, and so in our case we don't have to worry about that as much. But Jeff is absolutely correct in pointing that out. And we too would love to be able to redose. I think one and done is very theoretically appealing to many, to patients, physicians, and payers. But ultimately, what is most appealing is getting to the right effect. And so sometimes redosing may be important and necessary. So we keep our eye on that. What are the breakthroughs, and what are the, we were pleased with the recent Spark announcement that they had found a way to potentially enable redosing or evade the neutralizing antibodies. Frankly, as I shared at the top of the hour, we're modality agnostic. So, we work on gene therapy, we work on small molecules. I couldn't help but notice that Jeff doesn't yet have work going on in the heart. So that might be a fertile opportunity for collaboration to combine what we know about the heart with his technology. I think we have to have an open mind about what's the right solution to personalize the effect on a per patient and per therapeutic area basis. This is partly what makes us feel very exciting. But it's not going to be any one thing. I think it's going to be the collection of all these things that will allow us to make this transition to the broader therapeutic areas.
Yeah, just to chime in and echo on that point, I think the real opportunity is on combining orthogonal approaches, and I think there's many aspects that we're each focusing on, whether it be the vector or the payload or potential adjuvants you might give to address redosing or other things. But it's really combining those. And that's where the challenge comes in, because I think as companies, there's a limited set of things that you can focus on and do well. And we each kind of pick our areas where we want to kind of drive in. But then you do have to pick your head up every once in a while and see what's going on. And through collaboration, I think some really innovative things can get done. But it's going to take that. And we've seen that in other, new modalities. And you referenced one of those, Mike, in terms of antisense. It's when all these different approaches come together. But it all starts with, I think, really understanding how we can deliver safely and effectively, as that's been noted. But then from there, we can innovate and bring together multiple approaches in an interdisciplinary way. Yeah. Yeah.
Yeah, and so can you also maybe touch on pre-existing immunity and tissue tropism, some of the things that came up from both Jeff and Faraz, and how Stride Bio is approaching those opportunities. I mean, I don't know how many patients that are excluded from Luxturna or Zolgensma or one of the hemophilia gene therapies due to pre-existing immunity. But how much of an opportunity is just there if you have a novel capsid that avoids immunity?
Yeah, I mean, we certainly believe there's an opportunity there. That's the genesis of our company's foundation, which is to, as a first step, was to re-engineer capsids so that they would kick off these neutralizing antibodies that exist in patients. So the naturally occurring serotypes that are used for gene therapies, patients have been exposed to. And we can, through our structural techniques, determine exactly where they're binding on the AAV9 or AAV8 or whatever your starting point is, and then design around those footprints to limit that neutralizing antibody binding. And so I think it depends on the starting point in terms of the percentage of patients that are excluded from clinical trials. But I mean, it can be upwards of 40% depending on the potential starting serotype. So there's a big opportunity there. But then I think when you kind of peel the onion a bit, there's pre-existing antibodies, and then there's new antibodies that are going to arise once you do administer even a novel engineered capsid. And so, you're not going to stop patients from developing new antibodies. And so you have to address that as a separate approach as well. And so I think all of these things, what's great is that there's so many people working in the field and trying different approaches, and the data is just coming. And I mean, for those of you that go to ASGCT and kind of see the abstracts, I mean, the amount and volume of learnings is immense. And I think as a field, we're making a ton of progress. So I think a lot of these issues are tractable, particularly when you think about orthogonal approaches. But, the vector, I think, is part of the story and can address some of it. But I think you have to innovate around the other two areas in terms of, what you're delivering and then the process for manufacturing. One last point. One of the things we found at Stride Bio, we try to approach these things holistically. And what you've realized very quickly is if you try to focus on one particular parameter, let's say you want to make a virus that's incredibly potent, right? And then you're looking for engineering in high transduction efficiency. Well, then you find you can probably get to that. But guess what? Those viruses and vectors are so potent, they reinfect your producer cell line and you wreck your manufacturability, right? And so there's a give and take for this. And so you got to take a holistic approach and try and solve it that way. And Mother Nature has done a pretty good job with the parental serotypes. And I think we can improve it, obviously make better vectors. But you really need to understand that it's a tough nut, and you might improve one aspect and then find that you've sort of taken a step back on another aspect. So it's a challenging problem to address.
We have to be humble, Sapan. I mean, Mother Nature's had about a 2 billion year head start. So we're 20 years in the run. Yeah.
That's right. We're trying to supercharge our evolution, but no substitute for time and Mother Nature.
Right. Okay, so I mean, it's great. I have a whole panel of CEOs. As CEOs of gene therapy platform developing companies, one of the challenges that we've had is when we value a whole platform of say AAV gene therapy or some non-viral gene therapies that are going after ultra rare diseases and they're one and done treatments. It leads to very interesting revenue curves and thinking about how we're going to grow a portfolio. And from a CEO perspective, I guess you must think about meeting investors, investment desires, as well as pursuing the potential of your gene therapy platform. And you all have different, approaches to that, different business models potentially. If you have a mixed business model of small molecules and gene therapies and in vivo reprogramming, if you have a retreatment model, maybe you could talk a little bit, about that, as well as, if you can go into one of these big prevalent diseases, you have a very different, growth curve as well. So just, what are your thoughts and perspectives on, the changing sort of biotech business models to accommodate these different platform strategies. Who wants to? Faraz, you're grimacing when I brought it up, so you can start.
Smiling. The former commercial guy, it's inevitable that I'll think a little bit about commercial, but I also was schooled at Genzyme as Jeff was, where we were taught and rightly believed that if we do the right thing by the patients, if we follow the science and do the right thing by the patients, then the commercial rewards will follow. But we have to keep that end in mind. And so that's where, the things that we can influence now are things like manufacturability to bring down the cost of goods. And so we want to make sure that when we enter into the clinic for the product for a rare disease, and we think that there's upside potential to go from, say, a particular genetic form of dilated cardiomyopathy with a particular target, or from there we can see visibility to go into heart failure with a reduced ejection fraction, which affects millions of people. We're trying to make sure that when we've entered the clinic with that first asset, it is already with a manufacturing process that can scale up so that it can serve that larger patient population, so we don't have to go back to the drawing board. So we are thinking about commercial in that way from the beginning. One thing that I think we could all do a better job of as an industry is indication selection and portfolio sort of creation, for lack of a better word. It's not escaped, I'm sure anybody's noticed that there's a lot of crowding that's happened, right? We have multiple projects now in hemophilia, multiple projects in Duchenne muscular dystrophy, and there's a handful of diseases in which, sickle cell disease have multiple projects. Now, on one hand, that's great for patients. May the best product win. And so that's wonderful, right? That they have those choices. But on the other hand, I'd say for a new company starting today, they should think rationally about, it's tough to do rare disease recruitment anyway. Forget about the fact that rare disease recruitment, when there are 3 parties ahead of you with potentially one and done curative therapies, and that might be ahead of you in the clinic and ahead of you commercially, then you're chasing a diminishing pool at the end. So we've tried to be thoughtful, and that's why we've constructed the portfolio the way we have, with not only multiple modalities, gene therapy, regeneration, small molecules, but also orphan and prevalent, so that we have, I'd say, from an investor language perspective, multiple horizons of opportunity, that we might have some near-term shots on goal and multiple ones to be a fully integrated company that's launching its own products. But then behind that, there's other horizons with the larger opportunities that might take a more traditional phase 1-2, phase 2-3, or phase 1, phase 2, and phase 3 process to finally get to the finish line. I think portfolio selection is something that we could all do a better job of, and there's so much unmet need. I don't see the need for us to pile into the same couple of indications. That's just maybe my 2 cents.
It'll get reinforced for us when your girls start playing soccer. This is the way soccer goes for the first 10 years. They all face the same ball around. Can I add to your great intro there, Faraz? I think there's an aspect here too, which is allowing the horizon, which is visible for investors as it relates to gene therapy or genomic targeted therapies, to expand beyond small pools of allopathically focused Western countries to really global rare disease populations, which are generally speaking multiples of what we quote unquote can see or that are visible to us in the Western world. So that's one axis. And as the costs come down, that will become interesting, even commercially ultimately. But the question's not, to me, so much what will displace or disrupt or improve AAV. I think that's super important, and it's a worthy thing we're all chasing. Really one of the questions to ask ourselves is, how much bigger can biologics become? Because at the end of the day, any genome-targeted episomal or integrating therapy that produces a therapeutic protein can replace AAV, but can just as easily replace biologics. And if we're honest about it, the actual penetration of biologics globally are still minuscule. The largest biologics are probably treating about 200,000 patients worldwide. That's a tiny fraction of the number who could benefit from biologics. And of course, the capacity to make biologics is greatest inside each patient, not in a bioreactor in North Carolina or North Dakota or whatever, or Marlborough, Massachusetts. So I do think there's that opportunity to try to open up for investors, how large and how disruptive the impact of genome-targeted therapies ultimately can be when they scale.
Yeah.
Great. Sivan, you want to add to that at all?
No, one thing that resonates with me, I think Faraz mentioned it, as I think for the system to work, there has to be the presumption that if you come up with a treatment and meet an unmet need, that the system will find a way to support that commercially. But when you look at sort of the number of treatments, and as Faraz said, focusing on similar targets, there's a bit of a disconnect there because not all of those are going to be able to make it, and not even commercially, but in some of these areas, only one entrant's going to be able to get a clinical trial done in some of these populations, right? Because of the low numbers. So I think there's a fair amount of enthusiasm and optimism, but I think we take it on faith that sort of some of these things will get sorted out. But I think we all need to maybe do a better job earlier of thinking about how that's all going to play out. But the opportunity is so immense. I mean, I do think it's curious that, I don't know, Mike, you probably know these numbers better than me, but how many monogenic disorders, over 5,000 or so? But yet, if you do look to your clinicaltrials.gov list or other screen, everybody's focusing on the same kind of 20, couple dozen. So, I mean, I get excited about that, because, hey, there's a lot of opportunity there. And provided we can meet unmet needs, I think that, the systems on the commercial side will, I think, rise to the occasion. We need to do our part as well, though, and make them more accessible. So
Great.
Just my 2 cents. Yeah.
Great.
I was just going to make a final point. It just stimulates me to say it, but I mean, there is this incredible potential for modularity in the gene therapy space. So as delivery gets locked, as some of these elements of the construct become locked and these manufacturing breakthroughs, find their place, the opportunity to turn that wheel quickly, we're seeing that potential. I think we saw it first really with Alnylam's platform. I mean, the generativity of that platform now, 10, 15 years into the story, is extraordinary. And I think we're seeing something similar with mRNA. It's just good to remember how early we are in targeted therapies of the space. That's very promising. And I share your view that the 4,885 diseases that have nothing today, once you unlock delivery and you get the construct stuff modular, they should come quick.
Great. Okay, I think we're supposed to transition to audience questions. So Sarah, I don't know how this works if you pop on and ask questions, but I think we're happy to take some of the questions from the audience.
I think that sounds like a great idea. We've been getting quite a few, and they've been coming in literally from all over the world. And one of the questions coming from Nepal, actually, is how can we democratize gene therapies in least developed countries? How can we make it more affordable? And I know you guys touched on that a bit, but that's a pretty valid question.
I wouldn't mind taking a first crack at that. So first, I've been to Nepal, lovely country, lovely people, and great to know that there's somebody out there thinking about Nepal. And to Jeff's point, I think there's a great opportunity for one and done therapies. Part of the problem with more traditional conventional therapies, including biologics, is to the extent that you need a supply chain to provide that drug week after week, month after month, that gets tough for the infrastructure, the local healthcare infrastructure in developing countries. I've had the good opportunity to speak with physicians and government officials in different countries, including in the developing world, and the appeal of a one and done is high over there because they can actually wrap their head around it. If you can bring the cost to a reasonable level, and we've seen this with transplantation. I mean, transplantation, solid organ and liquid tumor and hematopoietic transplantation has been democratized and is being used around the world to cure patients, and it's one and done, and that the system can handle it. And a poor analogy for this is the way a lot of developing countries leapfrog past the era of wires and cables and went straight to cell phones, right? I mean, they overcome their infrastructure barrier and went straight to a technology that actually could work in the local context and did amazing things with that. So I view the one and done gene therapy paradigm as something that could enable better access and indeed democratize the access to cures. You could also do things like fly patients to centers of excellence in Western countries. And that, I mean, so part of what we have to do as an industry is think about our business model and widen the horizon here of opportunity and think, how would we make that happen? Through manufacturing, through logistics, through travel, but primarily through engagement. So we have to keep these kind of countries in mind, and we have to engage them at some level, not like 10 years after launch, but we should be thinking about that now. A great example was Bluebird with their thalassemia program. Their phase 1/2 study already was recruiting patients from Thailand, which is a country that has one of the largest prevalence of that disease in the world. Who would have thunk that, right? Most people would have focused in US and Europe, but they went all the way to Thailand to source patients. So I have great hope that we will reach Nepal with our therapies.
Wow, that's really great to hear. Really great to hear. On the other hand, we have another question that came in that relates to the cost in developed countries, or at least in the US, of the one and done of 2 million plus. How do you handle that?
Oh, go ahead, Sophan. Go ahead.
No. I was just going to echo some of the points that we made during the prior discussion. I think you're going to need innovation on several aspects, particularly around the manufacturing and reducing cost of goods. I think that'll be, a key part of it. The other, I think, frankly, when you compare those upfront costs and those numbers that are being adhered to, and you compare that to sort of the pharmacoeconomic costs of a lifetime of treatment, if we can find a way to spread out those costs and make it more reasonable and be creative around that, I think that's going to be helpful as well. It does, though, it's a very jarring upfront cost, but that has to do with a lot of factors on the expense of making the therapy and the R&D costs. But I think we can be creative about how we spread that out over time.
Okay, great. Thanks, Sophan. Another question came in, and this particular viewer is interested in knowing what your thoughts are with regard to DNA versus RNA gene therapies. What do you think the pros and the cons are of each?
I mean, I think they're both very powerful modalities, and I don't think there's probably a winner, right? I think all of these technologies, as Sophan was saying earlier, will find their place according to their relative strengths and limitations. I think broadly speaking, therapies that are active in the nucleus, DNA therapies, have the potential to take advantage of some of the regulatory infrastructure and mechanisms that reside there, which can be very long-lived. But as a balancing force, they're also highly complex and highly regulated, and there is probably such a thing as intranuclear immunity, if you will. And so lots to solve on that side of the equation. RNA-based techniques, on the other hand, they're pretty short-lived, and they have a less rich regulatory environment, but still they do have a regulatory environment. And I think what's emerging in that field is an unexpected degree of specificity in the toolkit, which I think can be incredibly powerful for certain indications. So I don't think it's probably possible to say that one is in principle better than the other. I think it's where the
yeah.
Anybody else want to comment on that?
I would just say, we're seeing it play out a little bit in SMA. Not only, I think you're speaking, Jeff, about augmentation, mRNA augmentation, but on the inhibition side or exon skipping side in that case, we're seeing a competition right now between a one-time curative gene therapy and a intrathecally administered RNA therapeutic in that case. So it'll be interesting to see the way that plays out, and that's just happening right now. And we also have a small molecule coming into the market as well. So it's a very dynamic space in something that was untreatable just a short period of time ago. Yeah. Another comment I'd like to say too is on the inhibition side again is that GalNAc targeted siRNA is showing very long duration of effect in liver-targeted diseases. And you know, it'll be interesting to see what happens to inclisiran and how it competes with PCSK9 antibodies, you know, once it's launched and has twice annual dosing. So, you know, and then what would happen if a PCSK9, you know, one-time gene therapy came into the space? You know, how would that play out? But
yeah. Well, just one other comment to make. I think, you know, we talk about these different modalities like they're black and white, but I think they're going to blur together. You know, as an example, I can take an AAV vector that delivers a DNA sequence that expresses an RNA sequence that silences another RNA sequence that unsilences a gene. And so, you know, all of these things can come together and have a positive therapeutic benefit. And that's what's exciting about the field. Everywhere you turn, there's something new that can be added to the mix for patients.
Yeah, building on that response, Sapan, another viewer asks if you can imagine, or if you are imagining that you will use this therapy to treat common disorders rather than rare disorders. Do you all imagine a day when that will happen?
It's part of the business plan.
Absolutely. I mean, look, we're here to change. We're here to change global health. I mean, changing the health of 1,000 patients can be extraordinarily impactful, and many of us have had the chance to see what that looks like up close. And the things that are really the scourge of humanity from the point of view of morbidity and mortality are common diseases. So I think in the same way that Faraz says we have to have global access in mind from the beginning of these company stories, we should also have prevalent diseases in mind if we really want to move the needle on human health.
Yeah, and I just say, like, heart disease is a great example where, you know, there's been more disappointments and failures recently than successes, but part of it is, you know, that, you know, it's still the leading cause of death in the world with all the innovation that we've had, more than all oncology combined, which makes it appealing and attractive. But we have to understand why so many therapeutics have failed. There's a lot of variability, comorbidities later in life, you know, obesity, hypertension, diabetes. And so, you know, part of our strategy is let's not just take that new science that we have and dive right into the deep end of that pool, but let's test drive that science in a rare population where the upside is it can work and save lives of patients with those rare disease. And then the even bigger upside then is you validated the efficacy and safety, and now you go into that larger population with more confidence. And so it's really not an either or. It's about just being thoughtful about how do we go after both in a way that makes sense for the company, makes sense for those patient populations.
Great, thank you, Faraz. One of our other viewers wants to know what potential do you see for non-viral delivery systems? Can are they potentially have advantages in manufacturing and packaging? Thoughts?
Yeah, I mean that's a little bit in our wheelhouse in the sense that we've steered away from protein-based delivery and really tried to leverage some of the incredible innovations that really started with Alnylam and have been further developed by Moderna and Translate and Intellia and others. I mean, lipid nanoparticles, I think, have a few features that are really attractive, one of which is that there is no pre-existing immunity and there is no adaptive immunity that is raised when they're dosed on a chronic basis, which we now have many, many years experience of in Alnylam's patisiran program. You know, as we think about the evolution to what we now think of as third generation LNPs, there is this opportunity to target them and to have them deliver with a much higher degree of tissue tropism than has been possible even for capsids in the past. So one of the things that's really beneficial as you think about the overlap with some of these global demands is that LNPs are really highly scalable from a process perspective once you've worked out the specific chemistry and elements that are fit for purpose for your payload. So they're really amenable to scaling, and I think they have that significant advantage over capsid-based approaches. So I think there's lots of potential, and there's lots to figure out. The LNP field is much more mature than it's been, and it's got a long way to go.
Okay, thank you. Thank you for that response, Jeff. Just for all of our viewers who are still submitting questions, we will, if we can't get to it during the live session today, we will work to try to get you responses. So don't Fred, if we can't get to your questions today. Another question has come in, gentlemen, from India, actually. The question is, what is the role of immunodominant antigen in vivo gene therapy, and how does it work to translate in the translation to therapeutical complacency?
Yeah, not sure I followed that, Sarah.
Okay.
Maybe repeat the question.
Yeah, what is the role of immunodominant antigen in vivo gene therapy and how it works to translate therapeutical complacency? Does that make more sense?
Yeah, maybe it's. I mean, I could take a stab. Maybe the question is around, inherent antigenicity for some of the delivery vectors that we're using, and as was highlighted by the other speakers, it is an issue that we're trying to address, and there are a few different ways. At Stride Bio, we're looking at actually engineering a vector that is able to evade those neutralizing antibodies, but there are other orthogonal approaches as well that could be effective. So we're trying to address that, but it is a limit to getting the right amount of vector to the right cells in a patient, for sure.
Okay, and I think this will, thank you for taking that on, Sapin.
Yeah.
And this will, I believe, be our last question, unless all of you really want to hang around a bit longer, since it's 12:06. This question is, has there been progress with the immune system counterattack on these viral vector delivery systems?
Another immune response question. So I think, there's two facets to it. One, there's pre-existing immune response, and then there's sort of the reaction to a novel protein that's being shown in the body, and so you have complement activation and other immune response. And so I think the field is trying to address both. It's an issue. And I think innovation around the vector, creativity around the payload that you're delivering, all of these things are going to be important to solve that. But we are seeing at high doses of at least AAV vector potential issues related to the body's immune response. So we do need to pay close attention to that.
Okay. Okay, thank you. Thank you very much. Does anybody else want to respond to that question? Mike, do you have any?
I would just add.
Go ahead.
Go ahead.
Oh, Mike, I was just going to say that we are at the beginning of a new era of medicine. And if you look back in time to other sort of transition points, whether it was the introduction of antibodies, decades ago, or then the orphan drug movement, there's going to always be scientific and technical, barriers to progress that need to be identified and overcome. And we're seeing a similar moment with the limitations of vectorology and geneticity. Some of these questions are around that manufacturing. And I think I have confidence in the human sort of innovative spirit that we will overcome. We will solve those problems. And if I would want the audience to take a step back and take away something, part of what made these transitions possible, part of what made antibodies successful, and part of what made orphan diseases successful in some way. At some level, you have to take a whole systemic view of things. And I think Genzyme and the orphan movement was a great example where the science had arrived, but the business model had to evolve. And I think that's sort of the similar moment that we're at here with gene and cell therapies or curative therapies, irrespective of the delivery modality, whether it's viral vectors or non-viral vectors. So thinking through the business model, what will make the future leader, the leaders of tomorrow, making good choices about business model, being nimble, being open, being modular, thinking with the end in mind. I think, and balancing that with being a specialist at what you're really good at doing. I think those will be the companies that will hopefully still be reading about 2 decades from now. That that company got it right at the beginning, and it wasn't just about breaking one particular problem down, like the neutralizing antibody or antigenicity, but solving the business model problem. And that was what allowed that technology to make that transition to large indications and become a relevant therapeutic modality. And I think that's what I want the audience to take away. We'll solve the technical stuff, not to diminish the importance of them.
I would add, the other hat that I wear is I work in immuno-oncology. And I would say we're learning so much about the immune system and the mechanisms behind detecting, self versus a foreign antigen on that side. And the adoptive cellular therapy field is editing and engineering each, receptor and pathway behind it. That we're learning so much about the immune system well beyond anything. We're designing and engineering molecules that, fend off, receptors that identify foreign, because the whole allogeneic cell therapy field is really dependent on developing stealth and cloaking, technology. So I think there's a lot that we're going to learn and cross over from cell therapy to gene therapy and vice versa.
Mike, I need to interrupt because I know, Jeff, you need to leave, right?
I do.
Okay,
I'm sorry,
okay.
Sharon. Yeah.
Okay. You can sign off. I'll do my wrap up. Jeff, thank you so much for being a part of this.
Thank you.
And we'll send you questions that are targeted for you and for everybody so that there can be some follow-up. Thank you very much, Jeff, for joining us.
Thanks, you guys. Really enjoyed being on the panel with you. Great day.
Okay. Great, and it's 12:10, so it's pretty much time to wrap up. And Mike, I want to thank you for moderating this session. It's a fascinating discussion that I think could go on for hours more, especially when you take a look at the questions that we've gotten in today. So thank you very much for doing that. Faraz and Sapin.
Thank you.
Gentlemen, thank you. Thank you for being a part of this.
Appreciate it.
As we sign off, I do want to, thank all of our sponsors, remind you that we will be continuing the virtual salon series. We're doing a pause in August, but we'll be back in September. And we hope to see everybody at BioFuture when and if we finally do get to the point where we can do an in-person event. So Mike, Faraz, Sapin, thank you. Our audience, thank you. Thank you, everybody who logged in from around the world. Bye now.
Great. And I just want to add, before we end, I remind everybody, I have a little bit of information packet on in vivo gene therapy. If you want, further information, please.
We can post it on our website. We can post it on the website, Mike.
Okay, great. Thank you.
For people to download.
Thank you.
Okay. Bye, everybody.
Bye-bye.
Bye-bye. Have a great day.
Good night, Nepal. Good night, India.
Yes. Good night. Good night, Europe. Good night. Yeah.
Yeah.
Bye.
Bye-bye.